Hello everyone, my name is Eugenia Fezza and I'm a PhD student at Harper Adam University. I'm currently awaiting for my VAVA and I've been focusing my research on Weevil, a pest of agriculture and ornamental crops. Today I will be presenting some key findings from my research and I'm excited to share this work with you and look forward to any questions or feedback you may have. The black weevil is a beetle belonging to the family of Curculionidae, characterized by brown 11 mm long body, black eyes, snout, and tufts of orange hairs on the fused wing cases. Adults are flightless and all the individuals are females with reproduction through parthenogenesis. Eggs are laid during the night below the soil and each female is capable of laying more than 3,000 eggs during its lifetime. Larvae emerge in summer are able to overwinter and pupate in February. We have usually one generation per year, but under favorable conditions, such as those in a glasshouse, the life cycle could be compressed so that more than one generation may occur. Whenever adults feed mainly on leaves of the host plants, producing this characteristic notching along the leaf margin. And while this notching has little impact on plant health, it can significantly reduce the economic value of ornamental crops where aesthetic is important. However, it is the larvae that negatively impact plant health by feeding on plant roots. Damage caused by weevil larvae can lead to complete crop losses if not effectively controlled. Control of weevil has largely relied on the use of insecticides, but due to the concerns on negative effects on environment and human health, the majority of them have been banned, so that currently there are no pesticides available for weevil. Nowadays, the only way to minimize weevil infestation is the use of entomatopathogenic nematodes. Despite the experimental efficacy of this control, growers continue to report significant crop losses associated with weevil infestation. It is argued that simply switching from synthetic insecticides to biological controls rather than use this control as a part of an integrated management program is a key factor in the continued importance of this pest. Here you can see a proposed weevil IPM. While our knowledge of weevil has clearly advanced, key knowledge gaps remain and making this IPM program difficult to be used. As a part of my PhD program, we aim to improve the sensitivity and reliability of monitor tools to detect the presence of adult weevil, identify behavioral active volatile compounds that may be used to manipulate weevil behavior, test registered botanical pesticides for use as weevil controls, and contribute to develop an effective IPM approach to weevil management. For this talk, I will mainly focus on the use of botanical pesticides as an alternative approach to control weevil. Botanical pesticides are naturally occurring chemicals extracted or derived from plants, and these phytochemicals have been shown to be effective for control insect pests by disrupting key physiological processes such as molting, respiration, as well as exciting the central nervous system. Phytochemicals have also been shown to modify insect behavior by acting as attracting, deterrent phagostimulants, anti-feedants, or anti-oviposition cues. And therefore, they represent a useful tool that could be incorporated into IPM programs. For my study, we tested Pitcher, which is a granular bioinsecticide based on garlic extract, commercially available in the United Kingdom for the control of weevil eggs and young larvae in ornamental crops. Laboratory bioassays were carried out to determine the toxicity of pitcher through contact and fumigation, as well as combined contact and fumigation on weevil eggs. Experimental units consisted of plastic containers lined with 2 filter papers. 30 weevil eggs were transferred to each container, and contact toxicity of pitcher to weevil eggs was determined in a direct contact bioassay. This was achieved by using, for the control experiment units, 3 milliliters of water pipetted into filter papers, while pitcher and 3 milliliters of water were added to the filter papers in the treatment experiment units. For the contact bioassay, the experiment units were left and sealed to minimize the potential fumigant effect of pitcher. For the fumigant toxicity bioassay, we used the same experimental units, but adding in petri dishes 3 milliliters of water in the control group or 3 milliliters of water pitcher inside, and these were put inside our experimental units to prevent direct contact between weevil eggs and pitcher. Experimental units were sealed with airtight lids. For the combined contact fumigant toxicity of pitcher to weevil eggs, we used the bioassay setup described for the contact toxicity bioassay, but also sealing the experiment units with airtight lids. In this way, fumigant and contact effect of the bioinsecticide could be determined. Results show that garlic significantly reduced hatch rate compared to the control group when applied as fumigant. Similar, the egg hatch rate was reduced compared to the control group when garlic was applied as a combined contact and fumigant application. No effect was observed when the garlic product was applied as a contact application. Laboratory bioassays were carried out to determine the toxicity of pitcher through contact of fumigant, as well as combined contact fumigation also on weevil larvae. Experiment units consisted of plastic petri dishes lined up with filter papers, and individual weevil larvae were added to each petri dish, and we tested, as we have done with the eggs, contact toxicity, fumigant toxicity, and the combined contact and fumigant toxicity of pitcher. The bioinsecticide significantly reduced the larval survival when either contact or fumigant application were used. We carried out also a combined contact fumigant effect to investigate the effect of this garlic-based product on weevil eggs exposed for 30 days in plastic containers containing growing media. And the number of larvae recovered after this period was significantly reduced compared to the control group. This study demonstrates the potential of garlic-based bioinsecticides such as pitcher to suppress weevil eggs and larval development in controlled conditions. The product multiple toxic effects, contact, fumigant, and both combined toxicity are particularly promising for effective control. As we mentioned before, IPM is defined as a strategy to combat plant pests, and this is using all available methods. Recent studies have shown that combining two different management strategies can result in improving pest suppression than when used individually. Botanical bioinsecticides are often used in conjunction with other IPM strategies, such as, for example, entomatopathogenic nematodes. This reduces the cost of biological control application as well as grower reliance of chemical insecticides. Initial research has shown that plant-based products may not affect the survival, virulence, or infectivity of entomatopathogenic nematodes and natural enemies when combined. To investigate the effect of pitcher on entomatopathogenic nematodes, we carried out this simple survival test where we put a fixed amount of entomatopathogenic nematodes on the pots with or without pitcher and growing media. We counted the entomatopathogenic nematodes present in the pots after 1, 3, 7, 14, 15, 21 days. We found that generally the number of entomatopathogenic nematodes over the time decreases, but this happens more in the pots treated with pitcher. However, after 21 days, still we can see some entomatopathogenic nematodes in the pots treated with pitcher. Despite this, we don't know if the entomatopathogenic nematodes treated with pitcher will be still able to infect, for example, Weevil larvae. To conclude, this study demonstrates the potential of garlic-based bioinsecticides such as pitcher to suppress Weevil eggs and larval development in controlled conditions. In addition, pitcher does not appear to be compatible with the tested nematodes when applied at the same time. Therefore, future studies are needed to determine whether the potential of garlic-based bioinsecticides can be realized as a sustainable Weevil management tool. I would like to thank my supervisors for the help and support provided during this PhD. And I would like also to thank you all for listening and for taking time to join this session. I am happy to take any question you may have.